Hello students, this is Dr. Faizan Mirza. We are discussing homeostasis. Homeostasis is the maintenance of a constant internal environment in a constantly changing external environment, which means the environment around the cell or around the organism is changing, whereas the internal environment with respect to its surroundings is being kept constant. The term internal environment and external environment can be considered at two different levels. For instance, the term internal environment can be used to refer to the environment within a cell or in an organism. In a similar way, the external environment in that case for a cell will be the environment around the cell, that will be its external environment, and for an organism, the environment around the organism will be its external environment. There are sensory cells that serve as receptors. They monitor and detect changes, and when they monitor and they detect changes, they are monitoring certain factors or certain variables or parameters outside the cell with respect to inside. Each parameter, each factor or variable is kept maintained at a constant set point, which will be in a narrow range. This is called as the set point of that particular factor. This brings us to stimulus. Stimulus is any deviation in the factor above or below the set point or beyond the normal range, either above it or below it. The stimulus, which is a change, it is sensed or detected by the receptors, which are the sensory cells. They monitor changes. They detect this change, and then restoration is brought about and the factor returns back to normal set values, and this is homeostasis. Example of certain factors, parameters, which are monitored at a certain set point in mammals are being displayed here. For example, the factor of body temperature. You know, the range is 37 degrees Celsius. In fact, it's a set point. Blood pH is about 7.35 to 7.45. This is in a range. Blood glucose is about 80 to 120 milligram per deciliter of blood. Similarly, your blood pressure is 120 by 80 millimeters of mercury. And even your ions are a factor independent of each other. So sodium ion in blood is about 136 to 145 millimole per liter, and potassium in blood is 3.6 to 5.2 millimole per liter. So every factor is maintained at a certain range. Any change in this range above it or below it will be detected by the sensory cells and the receptors to bring about homeostasis. This brings us to homeostatic control. This is the basic flow diagram of how it occurs. A stimulus comes which shows that the factor rises above the set point. Receptors sense this change, and the effectors then receive information from the receptor to decrease the factor back to the normal value. For example, if the stimulus was that the factor fell below a set point, in that case, the effectors will respond by increasing the factor, and this will be the response. So you can see that in either case, the stimulus, whatever the stimulus was, the response was opposite to it. If the stimulus was that the factor increased, the response was to decrease it. If the stimulus was that it decreased, the response was to increase it. This brings us to negative feedback, the loop through which homeostasis work. Mechanism of feedback that brings the factor back to the normal set value by working against the stimulus. This is called as negative feedback. Because the stimulus is a change, so negative feedback will restore it to the original set value. For example, if the blood glucose gets increased, you know that after the meal blood glucose gets increased, insulin is released, and the release of insulin cause the effectors, which are your liver and muscles, to take up excess glucose, and the blood glucose is lowered back to its initial set point of 80 to 120 milligram per deciliter. Negative feedback is always associated with homeostasis. You can't have homeostasis without negative feedback. On the other hand, positive feedback is not homeostasis. Positive feedback is also a mechanism of feedback, but that's not homeostasis. It's a feedback that takes the factor away from the set point by working to enhance the stimulus or the change further. For example, the entry of sodium ions inside the neurons or the muscles initiate depolarization. This is a change, this is a stimulus. It should be brought back to normal if it was negative feedback. But here, entry of sodium ions into the muscles or the neuron, it triggers the entry of even more sodium ions inside the cell. So this is no homeostasis, this is positive feedback. If we compare the coordinating systems, there are two coordinating systems in your body, endocrine system and the nervous system. Endocrine system works by sending chemical messengers, the hormones in your body fluids, primarily the bloodstream. Whereas the nervous communication deals with electrochemical actions which are sent as impulses and transmitted via neurons. The hormones, they are broadcast all over the blood, all over the body. They travel throughout the body in the fluids, and they look for their target tissues. Whereas action potential, the impulses in the neurons, they are targeted to specific cells, which will always be either a gland that would start to secrete something or a muscle that would end up contracting upon stimulation from the nervous system. Whereas the endocrine system, in most cases, it brings about a metabolic change in the effector organ or the cell. The effects of endocrine coordination can range from many minutes to several hours or even longer. And these effects tend to be mostly long-lasting, but not always. But yes, mostly they are long-lasting. If you compare them to nervous system, the effect is produced within milliseconds, within a fraction of a second. It's very quick and prompt. And the effects are a bit short-lived and reversible mostly, but not always. So that basically brings us to the end of the introduction of homeostasis. Thank you so much.